All right, everybody. We're going to get things kicked off here. Welcome and thanks everybody for joining us on our 12th episode of Ewings U. For those of you that don't know me, I'm Piero Spada, one of your co-hosts today, and also one of the co-founders at Little Warrior Foundation. But more importantly, I'm father to 12-year-old little warrior Lucy. If you're part of our core contingency, aka part of the Little Warrior Fight Club, and have been here since day one, thank you for making this a place of positive change. And if you're new, well, welcome to Ewings U. It's our webinar series where we hear from leading sarcoma experts and discuss the cutting edge of care and the science behind Ewing sarcoma. On that note, today we'll be hearing from one of the prominent experts in the field, Dr. Patrick Grohar, who wears two hats as both a clinician as well as a researcher in this field. Specifically, today we'll be exploring a key focus of his over the past decade, both his research and now open phase 1/2 clinical trials centered around the targeted agent, Trabectedin. Briefly, Dr. Grohar is an MD PhD and a pediatric oncologist and a physician scientist focused on the development of novel molecularly targeted therapies for pediatric solid tumors. After completing a Bachelor of Science in Chemistry at Villanova University, Dr. Grohar went on to complete both the MD and PhD degrees at Wayne State University. He completed pediatric training and fellowship in HEMONC at Johns Hopkins University and the National Institute of Health. He then held faculty appointments at Vanderbilt University and the Van Andel Institute prior to his arrival at CHOP as an associate professor of pediatrics and director of translational research within the Center for Childhood Cancer Research. And this next sentence I really like because it ties directly into this body of research. The Grohar Laboratory employs a bench to bedside and back again approach to developing targeted therapies for pediatric solid tumors. The laboratory has focused primarily on the therapeutic targeting of EWS-FLI1 transcription factor for Ewing sarcoma. EWS-FLI1 is the clear oncogenic driver of Ewing sarcoma, but also a challenging drug target. Their laboratory has employed a number of complementary approaches to directly target EWS-FLI1, develop targeted combination therapies, and identify novel drug targets. They have a number of studies in various stages of clinical translation. Moreover, Dr. Grohar is recognized for his work in Ewing sarcoma. He holds leadership positions within the Center for Childhood Cancer Research at CHOP and nationally within the Children's Oncology Group. He is vice chair of the Biology of Bone Tumor Committee, co-chair of the Combined Sarcoma Biology Committee, and chair of the Ewing Sarcoma Biology Committee. He is principal investigator with the Sarcoma Alliance for research through the collaboration, and he serves as ad hoc reviewer for multiple journals, the National Institute of Health, and for Cancer Prevention Research Institute of Texas. More recently, Dr. Grohar was appointed the Kelly and Chad Punchard Endowed Chair in Translational Sarcoma Research, an honor he is truly grateful for. In his free time, to which Dr. Grohar half-jokingly replied, "I have no free time," he likes to spend time with his wife and two kids, and he is an avid hockey fan and enjoys both watching professional NHL and moreover coaching his kids' hockey team. But before we get things kicked off with Dr. Grohar, I just want to let everybody know and remind you all that Ewings U is a webinar podcast production of the Little Warrior Foundation. We are a national childhood cancer foundation with a laser focus on finding a complete and permanent cure for Ewing sarcoma. We were founded in April of 2020 and have now granted out $1.6 million in high potential therapies for increasing survivorship and decreasing toxicity. And in order for us at Little Warrior Foundation to make programs like this a reality, we rely on the support of the Little and Big Warrior community. And we appreciate all of your support so that we can continue our mission of bulldozing our way towards more effective, less toxic therapies and continue this vital work for kids battling Ewing sarcoma. And before we hand it off, I'm going to introduce my substitute co-host, Hillary Lang. Hillary, thank you for being here with us.
Thanks for having me. Yep, my name's Hillary Lang. I'm a warrior mom to Ian, who he was diagnosed with Ewing sarcoma at the age of 15. He had a 4-year battle and passed away in 2021. During that 4 years, I researched or was forced to research quite a bit and also met the Spadas during that time. And we've kind of joined forces over the last couple of years. And I'm happy to be here. And thank you, Dr. Grohar, for being here.
Thanks, Hillary. All right, then one last housekeeping item. For all questions, please remember to use the Q&A box in the lower right-hand corner. It's just an easy way for us to field questions and help moderate questions. And please remember, we cannot case-specific questions. This is not the time, place, or format for that. And to be fair to Dr. Grohar, we got to keep these questions as general as possible. So please keep that in mind. And with that, I think enough from us. I'm going to pull my slides down, Dr. Grohar, and I'm going to let you take over.
All right, great. Well, Piero, thank you so much for the invitation to present, and thank you for the kind introduction. Let me go into presenter mode. All right, are we good? Are we full screen, or do I need to swap still?
You're good.
Okay, great. Well, Piero, I met Piero at ASCO, and he asked me to tell the story of SARC-37, which is a trial that's ongoing right now that, I'll ask the community, implore the community for help to complete. But I thought what I wanted to do was sort of place that story within the greater context of how I think about therapeutic development for Ewing sarcoma, and also how our approach sort of fits in, and maybe a little bit of history around this approach as well. These are my disclosures. All right, so I tend to think of Ewing sarcoma as sort of three distinct clinical needs with, sort of three distinct patient populations. Patients with localized disease, which I'll talk about in a minute, have, favorable outcomes on the most recent COG study, while patients with metastatic and relapse disease lag far behind. And so I think each of these subsets of patients has distinct challenges and a great need for improvement. So these are the Kaplan-Meier survival curves for the last COG study, in which the question was asked, could we add 2 additional active agents to the 5-drug backbone that many people on this call are familiar with to improve outcomes? And as you can see by these curves, there was no difference in outcomes for the experimental regimen, which included 2 new agents relative to the traditional 5-drug interval compressed regimen. So that was the bad news. The good news is overall outcomes were pretty good, actually, with 5-year overall survival up over 85%. Really quite a staggering number for this disease. So what's the approach moving forward for patients who are newly diagnosed with localized disease? Well, I think we as a community need to find who these patients are. Who are the patients who are high risk, and who are the patients that would benefit from more therapy, and what does that therapy look like? Because these 2 additional agents didn't improve outcomes. And who are the patients that would benefit from far less therapy? Who can we give less chemo to and shorten sort of that, long, difficult journey through primary Ewing's therapy? Now, it doesn't appear that clinical factors by themselves will be sufficient to delineate these 2 populations. Factors such as tumor size at diagnosis, location, age of diagnosis, soft tissue versus bony. So it's likely that we're going to need molecular factors coupled with clinical factors to determine who these patients are. Now, as many of the people on this call know, the Ewing sarcoma genome has been pretty well defined as many as 5 or 6 years ago. And it can be best described by the Circos plot. So this is a plot, if you're not used to seeing these types of things, a representation of all the different chromosomes and all the different mutations, such as copy number gains or changes, or somatic mutations or translocations. And in general, Ewing's is considered one of the low somatic mutation burden diseases. So in contrast to osteosarcoma, which has a ton of different mutations in a given tumor, Ewing's has a small number and is really defined by this chromosomal translocation called EWS-FLI. Now, it's been known for 25 years that there's a dependence on EWS-FLI, and this has led some of us to try to directly target EWS-FLI, which I'll talk about in a minute. But there are a significant number of disease-modifying mutations. And so the question is, do these disease-modifying mutations, do they confer the high-risk features that will help us delineate which patients are which? And so to answer this question, I organized an international consensus working group to sort of discuss what is known in the world of biomarkers that's ready to go right now for prime time to try to figure out who these patients are who are high risk versus low risk. And this included, patients from investigators from the United States, Canada, and across Europe, and many of the people responsible for the seminal observations that have characterized the genome. And we sort of defined the best candidate biomarkers that are available now, not necessarily maybe the best overall candidates, but the ones that are ready for prime time now, and published this paper with the hope that we will be able to analyze tissue retrospectively, and that's ongoing, and then maybe integrate it into upcoming COG and European clinical trials. But I think the real win for the community based on this work was that this led to the inclusion of Ewings in the CCDI molecular characterization initiative. So I don't know if people are aware of this, but as part of some of these moonshot funds from the federal government, there's been an expanded effort to profile pediatric tumors. And so some of the early tumors were like rhabdo and some brain tumors. And this led to like an explosion of information on molecular features of these tumors. And so we were able to make the argument that Ewings has a great need for this same exact molecular characterization. And it's been announced at several platforms that in the next upcoming COG study, all the patients will receive a multi-omic evaluation of the tumor, which will be a windfall for the community because there's not a lot of searchable, high-quality genomic data in Ewing sarcoma. And so this, I think, is a huge win for us. And we're working on approaches to build off of this, which if we have time at the end, we can talk about. But my laboratory focus for the last several years has been on this problem of relapse Ewing sarcoma. So as many on this call knows, many people on this call know, when a patient relapses, even if it's a small tumor at relapse, the outcomes are pretty poor. And this was captured in a review by Natalie Gaspar that was published almost 2 years ago that looked at the response rate of the last 62 published phase 2 studies representing over 800 Ewing sarcoma patients. And you can see from these numbers, the response rate is actually pretty poor. So if you look at the objective response rate, or so tumors that either completely went away or shrunk with therapy, the overall objective response rate for this 800 patients is only 10%. And of all of these studies, only 18% of these studies were considered positive. Okay, but what's worse than that is the metrics for positive was really poor. So a positive study was a progression-free survival rate of 4.5 months, or an overall survival rate of 16 months, or almost 17 months. So, clearly there's a need to improve outcomes, but this has led me to focus a lot of my efforts on, so why is that? Why are we failing to move drugs from, we've got all this great biology and all this great discovery science going on in multiple labs throughout this country and Europe and Canada. Why are these new agents and new molecules not being successfully translated to patients? Are we focusing on the wrong targets? Is it the wrong drug? Are we not optimizing how we're giving the drug to patients? Is it that by the time the patient relapses, this is a super highly selected sort of super clone of Ewing sarcoma? Or in fact, is it the wrong patient population to be testing new agents? Like by virtue of the fact that the patient has relapse, should we have more established agents that we are rolling out to serve these patients? Spoiler alert, I'm not sure what the answer is, but I think it's a little bit of all these things. I think we need to really focus on the context of Ewing sarcoma as its own thing. And then I think we need to do a better job of optimizing simple factors like dose, schedule, duration of administration of an agent, and model these in preclinical models. And so I'll tell a story, which I think is very emblematic of that. It's a little bit of all of these things. All right, so as Piero mentioned in the intro, my lab is focused on the direct therapeutic targeting of EWS fly from the time I was a fellow. So the first patient I met as a clinical fellow had Ewing sarcoma, and that's sort of driven me my entire career to work on targeting this molecule. I'm also a chemist by training, and so I reasoned, at the time, the mantra in the world was that it's quite clear that this is the oncogenic driver, but it's undruggable. And I was like, I'm a chemist, like everything's druggable. You just have to really figure out how to do it, okay? So I think, as I've said now a couple of times, this is like the perfect identity for a drug target. It's in every tumor. It's the only recurrent mutation occurring in like more than 20% of tumors, with STAG2 being the second most common. And the dependence has been demonstrated by multiple independent studies. So it's independent of one lab saying this is an important target. Like 20 labs have shown that if you turn off EWS fly, that's bad for the tumor and good for the patient. So why is it challenging? Well, transcription is complicated, and I'm not going to get into a long discussion of what transcription is, but I mean, essentially it's gene expression, and it involves like a very complex biology, which I've attempted to narrow down in just this simple figure, showing that it involves like decompacting of chromatin, exposing areas of DNA, and coordinating multiple large molecular weight complexes to direct the expression of specific genes at specific times, usually in development, but aberrantly in the setting of cancer. So where would you put a small molecule to inhibit this process? And, at the time when we started this, I'm not sure we totally understood how challenging this was going to be, because there were certain elements of the biology of EWS-fly that weren't understood at the time, but nonetheless, we charged headfirst into it, and I'll tell you a story of our approach. It is an exciting time for the therapeutic targeting of EWS-fly. So the first papers that were published of compounds that reverse sort of the transcriptional signature of EWS-fly were published between 2007 and 2011, so more than a decade ago. And now I think we're at the point now where many of these agents in the last 4 or 5 years have been tested in patients, and in the case of our studies are sort of on their second round through. And there is much ongoing work sort of trying to identify better and more specific molecules, which I'm not going to get into today. But the approach that we've employed in my lab is, as Piero mentioned in the introduction, a bench to bedside and back again approach. And so in many ways, this was sort of borne out by the fact that our first clinical trial of our first inhibitor didn't work. And that didn't make sense to me because the biology behind it was really strong. But we've used it to sort of evolve an approach where we use discovery approach to identify inhibitors, move them to patients as quickly as possible, and in the clinical setting, then characterize the limitations of the molecule. And then we can go back to the bench, refine the process, refine the combination therapy, and emerge with new combinations, second-generation inhibitors, and more detail about how to effectively administer these drugs. But importantly, this implies that the clinical translation is accompanied by sufficient correlative biology to both understand the limitations of the molecule, as well as what's different about the biology of relapse Ewing sarcoma, and I'll talk about that briefly. So I'm going to tell the story of Trabectedin today. And so this is a molecule that actually started in the clinic, and now we're on sort of our second loop around trying to make this compound work for Ewing sarcoma patients. I think this is sort of a busy slide, but I'll summarize it to say the critical thing about all of this approach is the recognition that the only way that compounds, I believe, are going to work in patients is if you understand how they work. If you understand how they work, then you can identify better, more highly tuned inhibitors like second versions and third versions. You can repurpose compounds for other indications based on common biology. And more importantly, you can more effectively move compounds from an observation that it stops cell growth to an animal, which I believe is the gold standard of preclinical work, and ultimately to patients. And then again, design clinical trials with the appropriate correlative biology to sort of inform the entire process. All right, so we became interested in this drug Trabectedin when I was a fellow. We had sort of been working on screens and approaches to inhibit EWS-fly. So we had developed all the assays to characterize whether or not you're inhibiting the target. And we sort of recognized that in the clinic, patients with Ewing sarcoma, there was a sort of a exceptional responder in a phase 1 study of a patient who had metastatic Ewing sarcoma, had a complete response to this drug. In addition, it seemed like of all the tumor types that had gotten Trabectedin, sarcomas were the ones that were emerging as the most sensitive to the drug in the clinic. And in fact, the subset of sarcomas that are translocation positive, such as Ewing sarcoma, myxoid liposarcoma, synovial sarcoma, were sort of the big winners with high response rates in patients. Now, myxoid liposarcoma had the highest response rate, and that's driven by a fusion protein called FUSE-CHOP, where FUSE is a family member of EWSR1. So this observation that, EWS-fly patients might be responding and FUSE-CHOP patients are responding, perhaps this drug inhibits the fusion protein. And so we published a paper now over a decade ago and showed that indeed it does. And we used a number of assays. We showed it, genome-wide using, at that time, microarray technology. We showed we could move EWS-fly into another cell context, activate a reporter, and rescue that with Trabectedin. And so the data was quite convincing, that the drug inhibited EWS-fly. Unfortunately, the following year after we published the paper, the COG published their clinical experience in 11 Ewing sarcoma patients, and there was only 1 patient with stable disease. And so again, I was really quite confident in our data. I'd actually done it with my own hands, and so it was quite clear that this was a disconnect. And so at that time, I reasoned that oh, it was probably schedule, because I could look at the drug exposure that we used in our preclinical studies relative to the drug exposure the patient saw, and they didn't match at all. And so I reasoned that, there's no way you're going to be able to get this back to patients if you just by saying, oh, well, the drug exposure was wrong, let's redo the study. Nobody's going to go for that. So in many ways, this led to the idea that if we understood how the drug worked, then perhaps we could administer it more effectively, potentially in combination with a potentiator, something that would amplify the effects that are on EWS fly. And so this slide just summarizes about 10 years of work in which we did, in fact, do that. We worked really hard to try to understand how the drug inhibits EWS fly. And so in brief, you can see here, this is a single EWS, single nucleus of Ewing sarcoma, stained green for EWS fly. And we would treat with the drug, it sort of moves it out of the neighborhood and concentrates it in an area of the nucleus called the nucleolus. And we could show using genetic approaches that this indeed triggered an epigenetic switch and led to chromatin changes at EWS fly target genes that led to a loss of expression. And we linked this to a specific drug exposure that was not achieved in the negative phase 2 and probably was achieved in the phase 1 study where I told you that patient had a complete response. We also published a paper of this synergy with irinotecan where we could show that trabectedin would sensitize Ewing cells to irinotecan, and then irinotecan could feed back and amplify the effects on EWS fly. But importantly, in any model that we tested this combination, the tumors just melted in the mouse. Okay, so these are called spaghetti plots. So each of these individual lines is the growth of a tumor in a mouse. This is tumor volume as a function of days of treatment. And so each of these is an individual tumor. And you can see in this particular cohort that we treated with trabectedin irinotecan in the schedule highlighted by the arrows, you can see that every tumor in every mouse goes away. And it didn't matter what model we've done this in. We've done, some of these are unpublished experiments, but in every mouse that sees trabectedin irinotecan, the tumor regresses. So I think that this is the gold standard preclinical result. Like I can't say it's the only thing you need to have, but if you don't regress a tumor in a mouse, you're probably not going to regress a tumor in a patient, in my opinion. The exception to that is every drug behaves differently in a mouse than in a person. So there are drugs that just don't work in a mouse that will work in a patient. So that's the exception to that idea. So we've opened this study, it's called SARC-37, which is a phase 1/2 study where we administered the drug as a 1-hour infusion instead of the 24-hour infusion that failed based on our preclinical modeling. And we combine it with irinotecan, and we've shown through a series of published papers that the irinotecan actually aids in the suppression of EWS fly, and trabectedin sensitizes to the irinotecan. These are the sites that we're open at, and these are our list of collaborators who are really many of the world leaders in sarcoma clinical trials: Leo Mascarenhas, Steve Du Bois, Bob Mackey, Ryan Roberts, Brian Crompton, John Glaude. Like it's really a Ted Leitch who did the infantile fibrosarcoma study, and Denise Reinke is my partner in crime in this. And so this is really a tour de force of sarcoma clinical trialists, translational scientists. Again, these are the sites that's been funded by the NCI, 1 million for Anna and Janssen. One of the challenges which we can talk about in Ewing sarcoma clinical trials is piecing together the funding to complete a study. I'm sure this group is well aware of that. But this is another example of, a study that we can make work with less funding, with the commitment of really great people. And so I presented these results at ASCO, and I just want to highlight a few things. So one is, we've completed now the phase 1. We're 11 patients into the phase 2. And in our phase 1 portion, we've established the dose and schedule. But some points to make. One, the median age of patients on our study was 18. We included a medical oncologist, Rashmi Chugh at University of Michigan, and this led to the inclusion of older patients, which are sometimes forgotten in Ewing sarcoma trials. But all of our patients had seen quite a bit of therapy. So the median number of prior lines of therapy of all our patients was 4. So most patients had seen 4 prior salvage regimens before enrolling on our trial, with a range of 2 to 9. In addition, almost the majority of our patients had previously seen irinotecan. We didn't think it would limit the activity of our combination, so we allowed them to enroll. And indeed, every patient that responded had already previously progressed on an irinotecan containing regimen. Our toxicities, this is still Trabectedin, so there was still a fair amount of toxicity. I'm not going to go through this rather busy chart other than to say it's still chemo and we have a ways to go to try to make it less toxic. But our approach has always been to sort of, let's get to efficacy first and then we'll worry about the toxicity. And so many of the approaches that we have going forward are trying to limit the toxicity that these patients are experiencing. But, it wasn't terrible. There was not a lot of nausea, vomiting. There's no hair loss. In general, patients had a pretty profound fatigue and almost feeling like they have like flu-like symptoms. And then these are the responses. So we really had, it's a phase 1, so we're dose escalating, and our first dose level wasn't active, but from dose level 2 and above, we saw a number of objective tumor responses. Okay, so if you're not used to looking at these as a waterfall plot, these are the best response of the patient on the intervals in which they were measured, where arrows going up means their tumors grew and lines going down means that the tumor shrank. And so at the recommended phase 2 dose, you can see these are highlighted by the asterisks. 3 out of 5 patients had confirmed partial responses. So almost 60% of the patients at the recommended phase 2 dose had a confirmed partial response versus the 8% to 10% that I presented in the last 62 studies. Limited patient numbers, early days of this combination, but really quite exciting, actually. And many of these responses were quite dramatic and led to quite significant clinical improvements. So there were a number of patients that came in with widespread bony pain that had really quite profound improvement in their pain with just 1 cycle. So 1 dose of Trabectedin. Here's a patient that came to the University of Michigan that was wheelchair bound and on oxygen with this huge lung tumor. After 2 cycles, you can see this thing shrunk by 40%, and the patient walked into clinic and Rashmi said looked great. Here's a patient at CHOP who had this 6-centimeter peripancreatic tumor that after 4 cycles was all but gone, and this patient went on to get 19 different cycles of therapy. So if this is the oncogenic driver of the tumor and we inhibit the oncogenic driver, this is what you would predict the responses would look like. Here's another patient who had this rather long pulmonary met that had a rather impressive regression after 2 cycles, and this is a patient who's currently on study with this rather dense pulmonary nodule that kind of becomes this wispy, almost transparent thing. And then lastly, we had patients that didn't meet criteria for objective responses, but here you can see this rather large lung tumor that after 2 cycles is calcified. So it's not going to shrink anymore, but it's it's quite different than in appearing than it originally was. All right, so this is really an exciting study. We've had a number of objective responses. We did also do some correlative biology, which I'll talk about in a minute. But in one particular example, we collected tissue pre and post treatment and can actually show that we can flip the EWS fly transcriptome. So genes that are induced by EWS fly are repressed with the treatment, and genes that are suppressed by EWS fly are induced with the treatment, consistent with the idea that we've inhibited EWS fly. I would say most patients at dose level 2 and above have benefited a lot we had many with stable disease, but a lot with rather impressive symptomatic improvement, which is a total win for patients and their families. In terms of the original questions that I would ask, this is a heavily pretreated population, and so you wonder about how this drug would perform up front, or this combination up front with a median of 4 prior lines of therapy and so many of these patients seeing Irinotecan. You would think that would have to have influenced the responses, even though that they were pretty good. And so I think the next step for this combination is to look at it either in the COG, or I've met with the European group that ran the Recur trial, and they're considering it as an arm in one of the next arms in that study, which we can talk about if you want. We did have a fair amount of toxicity, fatigue, myelosuppression, and liver enzyme elevations. This limited dosing in many patients, and so probably limited efficacy in some of those patients, but clearly is an area that we could improve on, and we're working very hard on a number of different efforts to improve that. Most tumors regressed, but eventually recurred, and so we need to try to understand why that is, but also we need less toxic alternatives. And so again, in my initial, circular bench to bedside and back, we are now investigating a second generation version of this drug called Lurbinectedin. So our preclinical studies have defined the fact that the threshold concentration of this drug is what really matters. And so this second generation drug accumulates to almost 17 times higher than the needed threshold in patients in the clinic. And there's some evidence of activity of this drug in Ewing sarcoma patients. And so we have a study that opens in the next couple of weeks, first here at CHOP, and then at really sort of the same group of sites. These are all the investigators that are involved, and Ted Leitch at CHOP is the co-leader, along with Bob Mackey, who's since moved to Memorial Sloan Kettering. And, again, just world-class sarcoma clinical trialists, Julia, Leo, Steve, Rashmi, and John will also be opening the study. And we've integrated patient advocates into this study. This is really going to be a strength, I think, in terms of accruing patients. And Carol Basso from 1 Million for Anna and Lori Carl from the Sean Carl Foundation are integrally involved in this study, which is really super great. And then, the last point I'll make is we've also layered in quite a bit of correlative biology, which I think will provide in both this study and SARC-37, we've obtained enough tissue to really ask important questions about what's the difference between relapse Ewing sarcoma and the same size tumor at diagnosis. And this includes, rather sophisticated transcriptome analysis, both on the single cell level and bulk transcriptome analysis, as well as pushing technologies such as like circulating tumor DNA and circulating tumor cells to try to understand the progressive, what is the difference between the progressive clone and those that are responding. And the nice thing is we've also built PDX models from patients. And so we'll be able to test our new agents in models that we know are resistant to this therapy in the clinic. And this is just kind of a teaser. Just one of the things we're looking at is, now we can profile the transcriptome of a single cell and look at, like, instead of one bulk analysis of the RNA signature of drug treatment, we can look at 10,000 different cells and what is the transcriptional differences in those individual cells. And so we've done this now in cell lines, and we can show there's quite a bit of difference actually from both model to model and within a tumor. And I think layering in some of the results from the clinic will drive many of the future directions that we take. All right, so I think that EWS-Fly is a viable drug target. And, I think we've had a number of molecules move into the clinic. And I think SARC-37 is going to end up a very important study. We've seen a number of striking regressions in phase 1, a trend that seems to be continuing in phase 2. And although tumors have shrunk, many of them have ultimately regrown and progressed. And so our approach moving forward is to identify more effective combinations, test them in the models that we've established in patients, as well as evaluate agents like Lurbinectedin, which already have an improved toxicity profile, and perhaps leverage things that are specific to EWS-Fly to develop novel, more targeted combination therapies. So maybe Irinotecan is not the best partner. Maybe we could come up with better partners that will both improve efficacy, but also limit the toxicity. I do think heterogeneity that I showed in that last slide is a fundamental feature of the EWS-Fly transcriptome and of Ewing sarcoma. And I think that we're going to have to learn as a community how to address this, both to understand relapse disease, but also metastatic disease. All right, and so, I'm really grateful for this platform because I think that this community can help. We need to finish this study, and we need to finish SARC-37 in a timely fashion. We only need to enroll 7 more patients, and enrollment has been very brisk. We only opened this study in, I think, January, February, and we've enrolled 11 patients. So please spread the word. I mean, this study we need to evaluate this combination. If it works, we need to move it to COG or to recur and determine whether this is an effective combination or not, and then move on to what's next. And so, yeah, we just need to finish it. Stand Up to Cancer, that study is open, but I think it's an example of some of the challenges of Ewing sarcoma clinical trials. Our reimbursements from that is terrible. We're reimbursing $3,000 a patient versus like an adult lung cancer trial, which probably reimburses $20,000 per patient. And so that means that all the sites that are enrolling our patients are going to have they're going to have to spend money to enroll a patient on a study, which really we should be able to at least support the infrastructure that a site needs to open up a patient. So I do think that this will negatively impact accrual. And so I'm working on trying to identify additional sources of funding. And the last thing I want to share with this community is, you notice I didn't talk at all about metastatic disease. And so we have not really sort of gone after this quite yet until now. So we've just launched a new initiative, which I'm not really going to talk about today, other than to say that this is really going to try to understand metastatic disease. It's really going to take a partnership between physician scientists, and the community to really collect high quality samples from patients with metastatic disease that will give us insight into what's different about metastatic disease versus primary disease, because I really don't think we really understand that in a manner that is going to lead to effective therapies in the near future. So we can talk about that too in the question and answer if you'd like. So many people to thank. So this is my lab. Alyssa Levine is my lab manager, who's done all the xenograph work. She's really a wizard, because these are really difficult drugs to give to animals, and she really cares for the animals in a tremendous way, which is important to our lab. Rebecca Kaufman is our bioinformatics scientist, who's done all the analysis of the work. And we have many great collaborators, both within CHOP, and I highlighted Ted Rowe is a great collaborator, and Leah Suri in pathology is fantastic, but also across the country. So the NCI, many people know John Glad and Brigida, Memorial Sloan Kettering. Within the COG, Katie Janeway and Damon are really supporters of this, even though they're not even involved in the study. And then Rashmi is my medical oncology colleague, who's really a tremendous partner in helping us to bridge some of these gaps. And we have wonderful collaborators at Memorial Sloan Kettering and the Dana-Farber as well. And we're fortunate to have obtained a fair amount of funding to support this work. So I'm very grateful to all of these funding mechanisms, and in particular foundations. So I want to thank you for what you do. It's something about getting a grant from a foundation which has been established by parents that's much more inspiring than getting a grant from the federal government, and kind of holds you accountable for your work. So thank you for your attention. I'm happy to answer any questions.
All right, Dr. Grohar, excellent job. I'm going to have Hillary kind of toggle through the questions, but I have just one basic question out of the gates. And it really speaks to what the mode of action of Trabectedin is. I kind of seen it listed as, is it a chemotherapy? Is it an epigenetic regulator? Is it truly targeting the fusion? Like, how do you, as an expert on this molecule, classify it, or is it multi-pronged?
Yeah, so I think it's a great example of a molecule that does a lot of things. So it's not a clean EWS-fly inhibitor. That's not exactly what it does. How it works to inhibit EWS-fly is it activates the DNA damage response, and it activates pathways that wild-type EWSR1 participate in. And so we're working on a paper now to show how, by activating the wild-type functions of EWSR1, we can impair the function of the fusion protein.
Okay. But would you call it itself a chemotherapy agent or not?
Yeah, it is a chemotherapy agent. It probably alkylates the microsatellites, although we haven't shown that quite yet. But again, it's the net effect, the cellular response to this particular agent doesn't happen with 20 other chemo agents that EWS-fly gets impaired with activity.
Right.
But by commandeering those and repurposing it in a rational way, we can inhibit the fusion protein.
Okay, thank you. Hillary, I'll let you take it away.
Sure. The first question, with respect to the spaghetti graphs of responsiveness, all mice had high positive initial reactions, but it looks like the graphs go up sharply after progressing to 0. What is the translation of those graphs post-tumor elimination?
Great question. So, one of the limitations of these experiments is, you can't, it's not like exactly like a clinical trial. So you don't redose the mice. Right. So the question was not, can we make the tumor go away and stay away? The question was, is it active? Okay. So if you look, can you still see my screen or no?
Yeah,
we can.
So if you look at like this experiment, the last arrow is here, and then we just want to see what the consequence is over time. So if we gave another cycle here, like we would in a patient, would the tumor stay away? I don't know. We haven't done that experiment, and it's limited by the actual administration of the drug. You can't continually dose an animal through the tail vein with this drug. It's just too locally caustic, and you can't put a port in a mouse. So one of the things that we try to do is we then recollect this tissue here at the upswing of the spaghetti plot and try to study that as a means of studying resistance. So I don't know if that answers the question. Was the question like, what is the translatability?
yes.
So I would argue that, in fact, this data shows that if you regress a tumor in a mouse, you can also regress a tumor with the same drug in a patient, right? At least the way we do the experiment. So we implant the tumors in a way that they're fairly aggressive in the gastroc. We let them reach a pretty, chunky size before we start treatment, and that really pushes the model to evaluate response. But it's just a model.
Gotcha. Can you comment on patients who have had prior brain metastasis and effectiveness of the trabectedin?
Yeah, we excluded brain metastases as an inclusion criteria. I know this is frustrating for parents looking for clinical trials, but the issue is if this drug has been associated with liver toxicity, and liver toxicity can be associated with bleeding. And so, as an experimental therapy, I don't think it's okay to put a patient at excessive risk without an established benefit. And so we excluded those patients.
Yeah.
I have a question, Dr. Grohar, before Hillary continues. How did you guys, what defined partial response for the clinical trial? I think I missed that in the slide.
Yeah, it's standard criteria. They're called RECIST criteria. Okay. Yeah. So you have to have a 30% decrease in the tumor on 2 successive scans. So for it to be a partial response, the tumor has to shrink by 30%, and then 2 more cycles, it has to stay shrunk by 30% for it to be a PR. So we had patients that we have unconfirmed PRs where it shrunk by 30%, but then grew prior to the next set of scans. And we had a patient that came down 29.7%. So that was a little disappointing as well, but it's pretty standard criteria.
Okay.
But it's pretty bad if you think about it. Like, we literally take CT scans and measure a tumor in 2 dimensions in 2023.
Right. Something that's 3D too, right?
Yeah, it should be better than that.
I have a question too, with regards to the spaghetti. So with the patients that you have now already in the trials, and you've given them several doses, are you seeing with each one it goes down more, or are you seeing that it knocks it down a little bit and then it's stable?
Yeah, so it's different in every patient, actually.
Every patient. Okay.
So, we have the one patient that it just kept shrinking and shrinking and shrinking and shrinking. And, the kid was on for 19 cycles, and then we had, of all the remaining lesions, only one, the ones were stable for a couple of months, and then one just started to inch up.
Okay. And then you have to go off the study at that point.
Yeah. And for that patient, well, anyway. So, and then we had other patients where it just didn't shrink much, but it stayed put for a long time.
Sure.
So I think it speaks to this heterogeneity of the tumor. Even if you make the assumption that you've inhibited EWS-FLI in all of these patients, you still see quite a bit of differences from one patient to the next, and really in the case of metastatic disease, from one nodule to the next.
Sure. And a lot of it has to do with the different mutations, is like STAG2. You don't know?
I don't think so, actually. We didn't see a pattern of, you would expect, so we profiled all of our patients, right, in relapse, which will be a really strength of this study, is that has not been seen before. And we expected to find everybody would have STAG2 mutations, but that wasn't the case. They didn't, actually. There was CDKN2A was our most represented, which is a known 12% of patients have that up front. And I think we had 3 patients of 18 or 19 that had the CDKN2A mutation, but we expected enrichment for these high risk mutations, and we didn't see that. What I think it is actually is that the transcriptome of EWS-FLI is going to be different from patient to patient, and that's where I think the answer is going to be. But I don't have any real data to support that idea.
Sure.
Do you want to comment on, I'm sorry, Hillary, while we're on the topic of biological correlates, I know you're in the throes of analyzing that and it's unpublished, but is there anything else you want to touch on there, or should we wait?
I think we better wait. It's still pretty early days, but I will say I did not see the pattern that I expected from our own work. We're okay, now that we've defined this, we'll have all this STAG2. So I think it speaks to the need to broaden the approach and make sure that we profile every patient from here moving forward. And I think the CCDI is going to help with that a lot for our next COG study, but we also really have to get a lot better at collecting tissue. And it is a staggering problem. I can't even begin to tell you how complicated it is. You would think that, if you think of an individual patient with this huge Ewings, you would think that we would have tons of tissue, but it's actually quite complicated. And I've worked a lot in the COG to try to improve it. And so this is going to lead to a new initiative to collect tissue that we're going to run outside of the COG, actually.
Are you gonna do whole exome sequencing there, or what, can you speak to that a little bit more?
For the CCDI, everyone will have DNA methylation, RNA sequencing, and whole exome.
Okay.
Awesome.
Wow, that's awesome.
It's awesome. Yeah. And,
That is
like,
awesome.
this is going to be such a boon for the community, because if you try to search Ewing sarcoma molecular data, like if you were a lung cancer researcher, you've got thousands of tumors you can sort of, work through ideas in silico before committing to any
Mhm.
laboratory investigation time. We don't have that. There's like
Yeah.
30 or 40 searchable tumors. Like, that's not good. Like, really, in 2023, that's not acceptable, especially when you've got these patients that come in with these huge tumors. Like, how does that happen?
Right.
And it's actually staggeringly complicated. And at CHOP, we've really focused on trying to collect tumors for specific histologies. And it literally takes a group of 20 people meeting every month, running the list of patients that have been diagnosed to not
Wow.
miss tumors. And even then you miss tumors, because there's a lot of different, it's not like leukemia. Everybody comes in through the ER and everyone has like an immediate
Yeah.
medical need. With Ewings, like, there's diagnosed out in out in a rural hospital. They start therapy because they think they can do it, and then they send them to CHOP. And it's just different orthopedic surgeons. Some are here, some are not. It's a huge problem, and I think it's absolutely going to take parents to help us figure this out.
Right.
Absolutely.
I think you kind of answered this one a little bit as far as timelines goes. It's very important, like you discussed, it's very important for response rates in relapse. The chart's showing like 16.5 months with the Trabectedin trial, partial response 60%, but what is the timeline?
Yeah, they're also progressing.
Okay.
So, this is refining again, what we went into this study thinking was we need to be able to regress tumors in Ewing sarcoma patients. That's step one. And I think we're doing that. Step two is understanding then why is it limited? And I think we're collecting the tissue that will speak to that somewhat. Certainly not comprehensively, but it's going to refine our approach. Like, that's a different question. How do you make a shrinking tumor continue to shrink, rather than, it's a different question than how do you shrink a tumor?
Right.
Okay, we're going to switch gears here a little bit. I'm trying to decipher this question a little bit. So it seems like that evidence of responsiveness becomes manifested pretty early in the treatment. Is there an opportunity to use specific patient tumor biopsy, presumably analysis in the lab, to understand what's affected before beginning actual treatment? I think this kind of dovetails with what you were mentioning of understanding the biology before we give treatment.
Yeah, I think that's an excellent question. Yeah, so there are approaches to try to screen molecules. They're called like hollow fiber assays, like screen a large number of molecules in patients. There's these avatar programs where you take a patient's tumor and you put them into a mouse, and then you see what they respond to. There's people looking at organoids as approaches to predict responsiveness. And then, there's good old-fashioned molecular profiling. So what is the profile of the responders versus the non-responders? I think the ideal would be that you could define a molecular profile, but more likely it's going to take a number of these approaches. Like, I think a lot of people are working hard on developing these organoid models where you could do a biopsy and screen molecules. I think that's a good approach. But yeah, I think that's an important question for sure. And really, really good question too. I don't know what the answer is. I think that there's a lot of different things people are trying, but
Hasn't got there yet.
hasn't got there yet. But I do think that, like, the most hopeful for me is molecular profiling, right? Because then if you can say, all right, this drug works and we have a couple of different agents now that I didn't talk about today, like one that's can't be 8q amplified, another one that can't be TP53. And, like, if you can start collecting, if we could better characterize our preclinical models and test our agents and put them in a pile, then I think we're more likely to have positive clinical trials. I think that this is, also particularly important for like osteosarcoma, for anybody who's interested in that tumor. Like that tumor is a genetic mess. And so, like, you're definitely going to have to characterize individual tumor molecular profiles to identify effective therapies.
Sure. One question was, can you say more about your new initiative on metastatic disease? Why can't the oncologist access the tumor samples many of us consented to at initial diagnosis? Project Child.
Yeah, great. So I'm trying to build a network to go after metastatic disease. The challenges are we often get a biopsy at diagnosis, but we don't ever sample multiple mets. And we rarely sample a tumor at diagnosis, multiple mets, and then when a patient progresses, we need that sample too. But we typically say, oh, that's Ewings, and then we go on with therapy, right? But what we need is cohorts of patients with a good amount of tissue with matched primary disease with a met at resection and then at progression. And like, that's going to take a commitment because the second biopsy of a met is not clinically indicated and puts a patient at risk. Okay, so that's the sample that we don't get because, it's not clinically indicated, right? So we need to get that sample or we're never going to understand what's the difference. And same thing at progression. Like if a patient had a primary localized disease and they relapse with multi-distant disease, what's different about those individual mets? Is there a difference or is it, one superclone that just is spread over? We don't have any tissue like that. I queried the COG bank for that collection of tissue and there were 3. Like that's not going to help. So with all the challenges to get biopsies, the ethical concerns that many people have about doing procedures that put patients at risk with no benefit to that individual patient, it's really going to take a partnership with parents because there's no actual benefit for that patient, but there's no benefit for the next 30 patients if we don't get that tissue and better understand the biology of metastatic disease in Ewings.
Have you tested a metastatic model in vivo in mice with trabectedin at all? Has that been done like with a lung metastases only to see, has that been done or not?
No, there's a couple really good metastatic models. Like Dave Loeb has developed this amputation model, which I think is really good, where it sort of mimics the clinical course of patients. Not everybody will do that model. That some people find ethical concerns with that. But we don't have a primarily genetically engineered mouse model, as I'm sure everybody on this call knows. So that's a problem. And yeah, the way that in the absence of those models, like, I think the model that we're testing is patients, which is not a real model. It's the actual real thing.
Yeah. It's frustrating, but I'm sure we're preaching to the choir here as a
very
clinician
frustrating.
Yes.
Yeah, very frustrating.
With this one, what is the recommended phase 2 dose for Trabectedin and Irinotecan?
It's low. Okay, so most people give Trabectedin. So by changing the schedule, we can actually give less Trabectedin, and then the Irinotecan is really low too. So most people give Trabectedin about 1.5 milligrams per meter squared. Ours is 1, and then for Irinotecan, people give either 25 milligrams per meter squared daily times 5 times 2. Some people do 50 daily times 5. We do 2 doses of 25. Okay. So we're seeing better responses with less drug, consistent with the idea that if you better understand how to administer a drug, you can at least take the next step in terms of broadening the therapeutic window.
And presumably less stomach issues with the lower dose and spread out, or same as usual?
No, didn't see one patient with diarrhea with our Irinotecan dosing. Not one patient had diarrhea. We prophylax, but like you prophylax with Irinotecan in any other protocol, doesn't really help. But so we didn't see one patient with diarrhea. It's a really low dose. If you think about it, it's only 50 milligrams per meter squared total versus 250, which was completely inactive in multiple studies by itself.
Interesting.
Yeah, it's the idea that like it matters how you give the drugs. We have a lot of combinations in the lab where we model extensively what dose to give first, what's the interval between drug A and drug B, what's the time that you need to maintain suppression, and that matters.
Yeah. So next question is from overseas. Is it possibly included in the study while staying in Europe? Do you have any European hospitals that are a part of this trial, or is that in the future?
No. So the problem with I have many great collaborators and friends that are in Europe, but the regulatory barriers to open this study on both sides of the Atlantic are not worth the time that you have to put into opening the study on two sites hurts the study rather than speeding accrual. So having said that, I was just in London for the annual Euro Ewing Consortium meeting. I gave a talk about this work and some other work. And they are they've come up with this awesome clinical trial backbone, which they call Recur, where they sort of tested 4 agents. I'm sure everybody's aware of this, and they got to sort of the winner, but and it was high dose ifos, which everyone was maybe like, really? But the important thing was it worked. It's now being used more here, and patients are responding. But the important thing is now they have like a response rate that they can just layer in a new agent and test it, or a new combination and test it against those response rates. And so I talked to him about maybe doing Trabectedin or Irinotecan as the next arm in the Recur portfolio. So it's coming. But we hope to get this published soon so people can use it based on the publication. And yeah.
Okay. Go ahead.
Do patients who have previously failed an Irinotecan based regimen have a decreased chance of response on this trial?
All of our best responders had previously progressed on Irinotecan containing regimens. So again, drug A sensitizes to drug B amplifies the effects of drug A. So the fact that they progressed on drug B didn't appear to matter.
Okay.
And for
Ewings,
you,
go ahead.
No, in Ewings that's important, right? Because everybody's seen Irinotecan by the time of clinical trials.
And then,
well, I want to kind of zoom out on that question too, because you kind of alluded to this, that it was a heavily pre-treated population already, right? Like 4 or 5 lines prior to starting this clinical trial. So how do we as a whole, like if this is a promising therapy, move towards getting patients into something like this as opposed to, the current plan B, which is just IT or another chemo? Like, are we there, or do we really need those biomarkers, you think, to kind of like classify which patients could potentially see a better therapeutic outcome?
Yeah, so I think, putting my COG hat on, I think the approach is IT is the standard in COG. So like all of our studies should be IT versus,
Right.
So once we have a promising agent like this, the next question should be, is it better than IT? So you do the side-by-side comparison and then answer the question. If it's not, then you stay with what is the current standard. And I think in many ways, that's the principle of Recur that the Europeans have, really so skillfully standardized. So I think that's it. But like, again, I don't know what that really means, because at the end of the day, a primary metastatic patient is not the same patient as a relapse patient. So if we develop all of our agents for this distinct population of relapsed, like, is it really applicable to the upfront setting to the patients with primary localized or primary metastatic disease? I don't think we actually know that.
Okay. I guess I'm just going to play devil's advocate right here from the patient population perspective. Like IT in a relapse scenario, overall outcomes aren't great, right? Like, I think you can agree on that, right? Like,
I totally agree.
There's a need to do better. And I guess, how do we just move faster towards getting there? And I don't know. It's a big question to unpack, and it's a loaded question, so I don't expect you to have like a clear pointed answer. I don't know. We just need to do better for these kids that really don't have viable options. So that's all.
I agree with you, but I think another way you can look at this story is, look, I published Trabectedin Irinotecan in 2014.
Yeah.
Right. So like, it can't take 9 years.
No.
To get your phase 1 cohort completed. It just can't. So like, aside from all the challenges of whether IT is the problem or whatever, like it has to be faster. It has to be more, cost effective or cost supported or whatever. I had to write 3 grants to get that study open and published 4 papers. Like, that's not an acceptable, that's not going to work.
Yeah, I hear you. It's maddening. That's all.
I like your bulldozing.
Trying to.
We need to bulldoze metastatic disease for sure.
Yeah. All right, Hillary, probably have time for like one more question if you want to ask one.
Let's see. There's a couple here. This one, would the oral irinotecan give the same outcomes as IV? Some studies show the same absorption for both, but sometimes IV is harsher on the side effects.
We went with IV. I felt like the exposure from oral is more like the published PK data on it is like a wider variability from patient to patient. So we went with IV. And we did see a fair bit of difference based on the tolerance of irinotecan. And so we added, looking for the UGT 1A1 star 28 polymorphism to the study. And so we'll report that as well. But I think patients do get a dramatically different exposure to irinotecan, and whether in this regimen that contributes to toxicity or activity, I think is something that we're going to have to figure out. That's an excellent question.
Last one, help us understand, how can we help as parents with tissue samples? This certain person has had initial Ewing sarcoma tissue at St. Jude. If she were to relapse, how could we get a tissue sample of the new tissues and also get that shared outside of St. Jude with other researchers?
Okay,
Basically, how can we help get tissue out there as
so
parents?
St. Jude is fantastic. So Beth Stewart, collects a lot of this tissue, shares everything that she gets, makes models, builds PDX models. So St. Jude, I think, is a bit of the exception.
Okay.
What I'm going to try to do is organize, so stay tuned, because we're going to try to organize parent groups to help with this problem, because you need to, capture patients at diagnosis, which, as you know better than I, is like an incredibly challenging time for a parent to be like, can we do an extra biopsy? Like that's almost like
Every parent would say, go right ahead, please.
Yeah, yes.
Is that right? Okay.
Yes.
Okay, that's amazing. So like I didn't know that. So we're going to work with that same Lori Carl, who I highlighted in as part of our stand-up study, to try to do some organization, and perhaps we can interface with this group as well to try to really get this tissue collected in a way that makes sense. Because I don't think you're going to be able to do it in the COG. We've tried many different ways. I don't think one institution or two institutions by themselves can do it. There's a lot of practical things that need to be supported, and funding, and we think we have a good amount of funding to support this. So please, if you're interested,
Would it be like a clinical trial that would be open for all sites that could that work, something like that?
So what I think, so the plan is, I said I wasn't going to talk about it, here we go. But the plan is, and we're still working out the details, so this is subject to change, but the plan is that you could piggyback it onto a COG study. So, there's going to be a COG metastatic study. Those patients are going to have tissue collected and going to the CCDI, and then you can piggyback with individual investigators at specific institutions that have, because it takes a lot of bandwidth, but a lot of institutions like CHOP, Dana-Farber, St. Jude have the type of infrastructure that you need in place to make this happen. And so can we leverage that infrastructure just by organizing with a little bit of support to collect this tissue? Because we absolutely have to do it.
Yeah, it needs to be organized for sure.
Yeah, you keep us posted on that initiative, Dr. Grohar.
Will do.
Yeah.
Will do.
Right.
Thank you.
We are running overtime, so I want to thank you, Dr. Grohar, for being here and for prioritizing our kids and sharing the story of Trabectedin. Like, this has been over a decade of research, and you're still at it, and we truly appreciate that. So thank you for everything that you do. And I want to thank my fabulous co-host Hillary today for being here with me. Thank you for filling in for Emily, much appreciated. And lastly, thank you to the viewers for joining us today as well. Again, we have a follow-up survey. Please give us feedback, how we can improve, what we're doing well, what we're not doing well, and what researchers do you want to hear from? That always is helpful. And we'll see you back here in August for our next session. To find out exactly when, please be sure to subscribe to Ewings U emails. Go to littlewarrior.org, navigate to the Ewings U tab, and you can also follow us on social media, both Instagram and Facebook, as we always post about our next speaker that way. And Dr. Grohar, I don't know if you know this, we have a sign off here at Little Warrior that embodies our fight in honor and in memory of so many little and big warriors who face this cancer. That's how we sign off on everything, internally and externally. It's just simply a 1, 2, 3 swords up. So if you would, the tail end of this, just give us a swords up. And Hillary, please join me as well, and we'll bring this session to a close. Truly did appreciate your time, and thanks again for being here, everybody. 1, 2, 3.
Swords up.
Swords up, everybody. Don't forget about
everybody.
the survey. And thank you.
Appreciate it.
See you next time.
Bye.